Hi all, good morning. I hope you all are doing well. Thank you for those who sent me an email to let me know that you're doing well. That I really appreciate that. So today we will be talking about transposition. We are still working on chapter 12, and this is the last lecture on DNA recombination. So transposition is part of DNA recombination. We've been talking about this for last couple of lectures. And then today I will also move on to the next module on transcription. So that will be a separate video. Okay. So we were talking about transposable elements. We talked about three major families of transposable elements: DNA transposons and two type of RNA transposons or retrotransposons. These retrotransposons can be virus-like retrotransposons, also known as LTRs or long terminal repeats containing retrotransposons, and poly-A retrotransposons. These are non-viral retrotransposons. And we were talking about different mechanisms, how these transposable elements jump from one place to another place in the genome. So again, just to remind you, these are three different types of transposable elements in the genomes of both prokaryotes and eukaryotes. The DNA transposon, they all have somewhat similar structure. They all have these terminal inverted repeats and these central sequence that will code for transposable element. So this is an autonomous transposable element that contains everything the transposable element need. Virus-like retrotransposons have these long terminal repeats. They also have these terminal inverted repeats that makes part of this LTR. And these also have the sequence that will code for integrase and reverse transcriptase. Poly-A retrotransposons are a bit different in structure than DNA transposons and virus-like retrotransposons because they look more similar to a gene that will code for an mRNA. They have a 5' UTR and 3' UTR and also contain two ORFs, open reading frames 1 and 2. Open reading frame 1 codes for protein. Those are RNA binding proteins, and ORF2 code for proteins that will make again the integrase and reverse transcriptase. They also contain these poly-A tails, and that's where the name comes from, poly-A retrotransposons, and that's quite important for integration of these transposable elements that we will talk about today. In the previous lecture, we talked about the mechanism of transposition. There are two major mechanisms of transposition: cut and paste and replicative transposition. So we're talking about DNA transposons here. Cut and paste is common for all different types of transposable elements. And then today we will talk about replicative transposition as well. Okay. So DNA transposition by replicative mechanism is it's a bit different from cut and paste that we discussed earlier. In a replicative transposition, instead of the transposable element just moving from one place to another place, going from a donor DNA to target DNA, just removing itself and going there like in cut and paste, it will copy itself into the target DNA and it will also stay in the donor DNA. So this leads to a net increase in the genome size that we talked about. So each round of transposition, the DNA will be duplicated, element DNA will be duplicated, and will it go insert itself into a different portion of the genome. The transposase protein assemble on the two ends of transposon. So here on these inverted repeats, that's where transposable the transposase proteins will assemble, just like we talked about in cut and paste. And this structure is now called transposome. Once those transposases bind to both inverted repeats, one here and one there, and then it will bring it together. You might want to look at your previous lecture slides for that. And then we talked about how this cleavage happens. It's the first portion of those transposases will cut to make the 3' end cut. They will result in this 3' hydroxyl here. They cut on both ends, both 3' ends, and these 3' hydroxyl Unlike cut and paste transposition, where the transposon is excised either by the action of this hydroxyl group will make a break here, and this hydroxyl will make a break here, or a different set of protein like we talked about in TN, I guess that's TN7, where the TNSA protein makes the cut. So in the cut and paste, it will be completely excised or removed, but in replicative transposition, that's not the case. It only makes these two cuts, and this hydroxyl group then will make a cut in a target DNA. The 5' end will stay bound to the donor DNA, whereas this 3' end now will bind to the target DNA. So here, this is the target DNA, this is the donor DNA. The donor DNA is still bound to the transposable element on one end, and the other end this binds to the donor DNA. Here again it makes this gap. We will talk about this in the next slide. This 3' hydroxyl end of the transposon, they joined to the target DNA site by the DNA strand transfer reaction, the recombination reaction. And that's where the recombination comes in. These are 3' hydroxyl groups here. These will make a strand break in the target DNA. It will make this DNA strand transfer reaction. So these are combined now. These two DNA molecules are now recombined. They're attached together. Makes this replication fork right here. This replication fork assembles at the left gap, and then from there, once this replication fork is formed, this transposable element will be replicated into this new sequence. So here the intermediate is generated here in the DNA transfer. It's a double branched DNA molecule where the 3' end of transposon is joined to the new target site, and 5' ends are still joined to the old DNA, the donor DNA. These two branches now within this intermediate, these look like a replication fork. So this will now recruit the replication proteins from the host cell. They will assemble on this fork. This 3' hydroxyl end will serve as a primer for DNA synthesis, because the synthesis is 5 to 3', so this will lead to that, this will act as a template, so this will now be replicated. So here, so this will now sequence, this will move in this direction, and finally this replication is carried out by polymerases, and then ligase will fill the nicks. So at the end you have your previous sequence, your donor sequence that had the transposable element. This transposable element is still part of that DNA molecule, and it also part of now this DNA molecule, so it's co-integrated with 2 copies of transposon. So again, this is a little better image to see. This is the transposable element right here. These are the inverted repeats. These are the sequence that makes the transposase. There's a 3' hydroxyl, free 3' hydroxyl group on each side. This 3' hydroxyl group will make a cut in the new DNA. This new and old DNA will now recombine to look like a replication fork for the cell, and cell will recruit replication machinery here, and this replication machinery will then use this replication. It goes from here to here, copying this sequence, from here to here, copying this sequence, and at the end it's both have the element has replicated to make 2 copies. These type of transposition, these replicative transpositions are quite detrimental for the cell, because these jumping genes, these are autonomous genes. All kind of transposition are detrimental to cell, but here it's not only it's moving from one place to one place, but it also changes the genome size. Now let's talk about the retrotransposons. There are 2 classes of retrotransposons: virus-like retrotransposons and the non-virus-like transposon, non-viral retrotransposons. These virus-like retrotransposons, they have a bit different mechanism, because these are retrotransposons. So they first make RNA molecule from the DNA molecule. So remember here, this is a DNA now. It's inserted within the DNA. This is the LTR, or long terminal repeat. It's long terminal repeat. They also have the inverted sequence within the long terminal repeat. The inverted repeats here on the 5' end also has a promoter sequence. It will act as a promoter. This promoter will initiate transcription of this element. So this is the whole element from here to here. This is this when you see this arrow, this always means that the transcription start site. So this is the transcription start site. So this is the promoter right here in this inverted repeat. This transcribes an RNA molecule. This is element RNA. This RNA molecule, it's an RNA intermediate. This RNA intermediate then reverse transcribes to form cDNA. Remember, these LTRs have a sequence that make both integrases as well as reverse transcriptases. So this reverse transcriptase here will reverse transcribe this RNA molecule into cDNA. So here's one copy of cDNA and then complementary copy of that cDNA. And this now it's double-stranded DNA molecule again. Then integrase, the other sequence that it codes for, that integrase will now catalyze 3' end cleavage. So here it removes part of this 3' end, similar to how the DNA transposon does the same thing. It breaks, it makes a cut at the 3' ends. Now these open 3', so let me do that. This retrotransposon sequence first transcribe RNA by RNA polymerase. These RNA polymerase, because this is the promoter sequence right here, this promoter sequence will assemble the RNA polymerase machinery. So RNA polymerase will synthesize this reverse transcript, will transcribe this RNA intermediate. This RNA will then reverse transcribed to generate the cDNA molecule here. So let's go to the next one. Then integrase or transposase will recognize the cDNA for recombination with new DNA sequence, target DNA sequence. Here the integrase makes these cuts at 3' to make these 3' hydroxyl group. Usually release a dinucleotide, and this dinucleotide release that's what makes that direct repeat here, or duplicated repeat here in the DNA, because it's going to go fill this sequence. This integrase assembles at the end of the cDNA, cleaves a few nucleotide as from here, makes these 3 hydroxyl groups. And then integrase will catalyze the integration or insertion of these cleaved 3' ends into the target DNA. Once this happens, then the gap will be repaired by the host's repair machinery, and it will fill the gap by our DNA polymerase, and ligase will fill the next. And now this has been inserted into here. And this is that the duplicated repeats, the direct repeats that transposable elements create in the DNA molecule wherever they insert. Poly-A retrotransposon, or non-viral retrotransposons. Another thing I wanted just to mention here is, so you can see here how they are structurally very similar to viruses, and that's why it's believed that these used to be viral DNA that has now been integrated into the genome and became the part of the genome. Poly-A retrotransposon, so these they use a different mechanism than virus-like retrotransposon in order to transpose from one place to another place in the genome. The mechanism they use is called reverse splicing mechanism, and as I said, it's different from the other retro elements. This mechanism is also called as target site primed reverse transcription. So this is called target site primed reverse transcription. And you will see how it's different from the virus-like retrotransposition. So here, let's see this example of this line, this long insert nuclear element. this in this example here, so we have because these poly-A-reto transposons, remember their structure is similar to mRNAs. They have 5' UTR, 3' UTR, 2 ORFs, and poly-A tail. These elements, this 5' UTR contains the promoter sequence. That's where the transcription will start. So again, this is the transcription start site. This is the promoter sequence and 5' UTR. The first step is transcription of DNA of this integrated element by an RNA polymerase. So this RNA polymerase will start here, the promoter sequence, and will make this mRNA sequence, or will make this RNA molecule. So this is the line mRNA now that's transcribed from this DNA molecule. This is 5' UTR, again 3' UTR. This is the poly-A. This line mRNA will go out to the cytoplasm first and will be translated. It will make these two proteins, one coded by ORF1 and one coded by ORF2. Remember, ORF1 code for the protein that's RNA binding protein, and ORF2 code for proteins that are integrases and reverse transcriptases. Both of these proteins will bind to mRNA. So ORF1 will help bind both of these protein to mRNA, and this mRNA will come back to nucleus now. Promoter is embedded in the 5' UTR, so that's where this transcription will start. So here now you see that in the it's a different mechanism here, that the DNA is transcribed into mRNA first. mRNA goes out to cytoplasm, translated, make these proteins then again bind to the RNA, the same RNA molecule that makes those protein. This protein will bind to this mRNA molecule, and mRNA molecule along with these protein will come back to nucleus. Okay, this once this come back to nucleus, then these endonuclease will initiate the integration reaction by introducing NICs. So and now what happens in this poly-A tail helps determine where to go and insert itself into the genome. So most of the time these poly-A-reto transposons will integrate into the regions that have multiple T's in the sequence. So here this target DNA has this multiple T's. These multiple T's will bind to the poly-A tail. So this gives it a complementary sequence right there. So once this binds to this complementary sequence, this T-rich sequence is preferred cleavage site. So it will go that integrase, that protein. They have this integrase protein. That integrase protein will make a cut. Right? This gives this open 3' hydroxyl. It will make this cut, and then it will bind to this T-rich region. Now remember that it's still mRNA, still RNA alone attached to these proteins. So it's a complex of RNA and proteins. It's not DNA yet, but it's inserting itself into the DNA molecule, so it needs to be a DNA molecule, right? So it's a target site cleavage. RNA DNA hybrid is formed here, and at this RNA DNA hybrid that's where the synthesis of first cDNA strand occurs by the reverse transcriptase activity of this ORF2 protein. This reverse transcriptase activity of ORF2 will make this copy of this cDNA here. So it uses this because these A's and T's are now bound. It uses this sequence as a template, and it will make that sequence, will copy that sequence as a cDNA. Once the cDNA is formed, then RNA is degraded. This RNA degradation and second strand synthesis happens then at this point. And this RNA, once this RNA is degraded, second strand of cDNA is synthesized. Now the ligase will fill the nick here as well. And now you have this sequence inserted into a new region where it was a T-rich molecule. So many of these poly-A-reto transposons are detected in large-scale genome sequences, are truncated elements. What that means is most of these molecules don't have the 5' UTR. So if they don't have 5' UTR, that means they cannot transpose. So they're frozen elements. Because if it misses the 5' end, it does not have the intact promoter, and it does not have intact promoter, it cannot make that RNA molecule. If it cannot make RNA molecule, then it cannot transpose. So we talked about these long interspersed nuclear element or line, and we also talked about sign, short interspersed nuclear element. Signs are usually these frozen transposable lines where they lack the 5' UTR region. So here, these are the major types of transposable elements that we have talked about so far. DNA-mediated transposition is carried out by DNA transposable elements or DNA transposons. There are different examples of these bacterial replicative transposons, for example TN3, and we talked about bacterial cut and paste transposons in the last lecture, TN5, TN10, and TN7, depending on what kind of cut they are making. There are different kinds of different examples of these bacterial transposable elements. When we talk about eukaryotic DNA-mediated transposons, P elements of Drosophila is one of the most commonly used eukaryotic transposon. HAT members, examples one of the examples that we talked about, NTC1 or Mariner elements, these are also part of this HAT family. Anyway, as the T comes from the TC. RNA-mediated transposition is two different kinds, virus-like and non-viral or poly-A retrotransposons. These virus-like transposons have large, long terminal repeats up to 250 to 600 base pair. You remember in DNA-mediated transposon, those inverted repeats are maximum 100 base pair. Is usually not even that long, but the longest is about 100 base pair. So these long terminal repeats are much larger than the inverted repeats of DNA transposons. The example of these virus-like retrotransposons are tie elements or copia elements in Drosophila. These are not as commonly used as the P element. Most of the work that you will read in literature, transposable elements based transgenic, you will probably find more of the P element or Mariner elements, some copia elements as well, not too much of tie elements. Polyerythrotransposons we just talked about, these have these AT-rich sequence, 5' UTR flank, the encoding regions, that's where the transcriptions start. The promoter region is in 5' UTR. Example of polyerythrotransposons are F and G elements in Drosophila, line and sign elements in mammals, and LU sequence in humans. And these LU sequences, these are polyerythrotransposons, they make a pretty large part of human genome. So just to show you just an example of type of transposable elements in the human genomes, the lines, these are autonomous. That means they can transpose from one place to another place. They have everything they need. The copy number is about 20 to 40,000. They makes about 21% of the genome. Signs, the short interspersed nuclear elements, these are non-autonomous. As I mentioned, most of these lines lack 5' UTR, and these signs do not lack that 5' UTR of lines. They make about 13% of the genome. DNA transposons, these can be autonomous and non-autonomous as well. Autonomous means they can move from one space to another space. They have everything they need. And the non-autonomous is they lack the sequence that will produce the integrases or transposases. So if it lacks that sequence, that it will not be able to transpose unless there is a sequence that is very similar to these sequences and can make it move. Make about 3% of the genome. So this is just an example. We still have here we're not talking about the virus-like retrotransposons. Example of those, LO sequence. All these together make about 50% of the genome. And with that, this is all about transposable elements that we're going to talk about. Next time I will be moving on to transcription. Okay.